I got a couple pieces of news and then we're going to get going on the lecture. The first piece, and I'll say this again on Friday and on Monday, just because next Wednesday, Thanksgiving break officially starts at noon, but I will not be teaching well, I won't. You guys can come, but I'm not going to be here Wednesday morning. I used to do it in the past, but there was like 10 people in the classroom, and basically I just had to teach the same lecture again on the Monday after break, so we don't do it anymore. We're not supposed to do that, but it's not worth it. The second thing is that after this lecture, I'm going to be changing up the lectures significantly. I'll give you guys a heads up on what thing to download if you're bringing in notes and things like that, but don't follow the calendar directly. Content-wise, it's all going to be the same. I'm going to be adding some stuff, but I want to present it a little bit differently in a different sequence with different materials. Okay, so just be aware that that's coming down the pike. Okay, all right. So what I like to do is finish off this component of nutrient management and fertilizers, and this dynamic environmental dynamics and quality. I think this actually might be a good title for what's going to happen for the rest of the semester. We're going to be talking about this from a lot of different perspectives, from pollution perspective, from a remediation perspective, from a management perspective, from a measurement perspective. So I think when I say don't follow the calendar as to the lectures for the next, I guess, seven lectures left, something like that. Don't follow it, but this is basically what the content's going to be. All right, questions? We good to go? All right, so we're going to be talking about nutrient management. And when we talk about nutrient management, these are the things that you have to keep in mind when you're thinking about how nutrient cycling is working, how you're going to be managing issues of nutrient loss, nutrient gain. You're going to have to be thinking about the mineralogy, the parent material. Okay? You're also, in relationship to that parent material, you're going to have to be thinking about texture. Now, texture obviously has this issue with surface area. When we start talking about texture and parent material together, we're starting to talk about CEC and AEC, so exchange complexes. Okay? We also have to think about the climate. The climate is going to control, specifically for nutrient processes that are controlled by biological systems, certainly is going to control water, the amount of water, and the temperature is going to control the distribution and activity of organisms. But water and climate also have to do with, or temperature, water and temperature also have to do with the the chemical reactions with or without biological mediation. Okay? Biology, obviously related to climate, but the type of organic matter in the biota and what's going on there is certainly also going to affect how these nutrients are moving through the system. pH, solubility of nutrients. The solubility of these elements is really dependent upon the pH. And then in turn, this pH has a huge effect on exchange complexes. So the retention of those nutrients and how those nutrients move through the system. And then finally, the nutrients themselves. Okay? So today, we're going to talk about availability, nutrient availability. Okay? What is it? It is a vague term. Okay? What is it, though? Okay? Well, when we talk about nutrient availability, we sort of talk about three things. But most people think, well, how much of it is going to get to the plant? Okay? Well, how much of it's going to get to a plant has a lot to do on what is the concentration of that nutrient in the solution. Right? And it makes sense. Okay? Then, once that nutrient's gone, what's the speed of replenishment of that nutrient back into that nutrient solution? And then finally, when it's in the solution, how fast is it going to be moving through that system? Okay? Last lecture, we talked about the issue of mass flow versus concentration gradients. Okay? That has a huge impact on this mobility. And the mobility of the solubility of those nutrients or those ions in turn have a huge effect on the mobility. They're playing back to each other. Okay? The definition is the ability of a soil to maintain high concentrations. It's not really the ability to maintain high concentrations, but a fertile soil, in essence, is its ability to maintain high concentrations in solution and in the vicinity of the root. If the nutrients are in high concentration and they're not near the roots, they're not going to do a lot of good for the plant, right? Okay. Now, another definition we should talk about is this definition of what fertilizer is. Generally, people, when you say fertilizer, most people think of it's an inorganic material that you buy at Agway or something like that. Okay? A fertilizer is a material, natural or synthetic, inorganic or organic, that provides useful, notice that it's underlined, useful quantities of plant nutrient in a form that can become soluble in the soil. And in fact, it can be soluble already or can become soluble. Okay? It's dependent upon, or its availability is basically dependent on 3 factors. And those factors are the ones that we talked about already, these factors. Okay? And the fertilizer, in essence, also builds up the reserves to provide useful quantities. Okay? The definition is really this component of it. Now, that availability can vary from time to time and from soil to soil and from condition to condition. Okay? Availability may vary from soil to soil even when that same amount of nutrient is present in each one of those soils. Now, why? Well, it depends on those 3 factors again. The concentration of those nutrients in the solution, the speed of the nutrient replacement, and finally, the mobility of that nutrient in the soil. Sorry, I skipped back. I know you guys hate it when I do that. Okay, keep those 3 things in mind. Some of the nutrients can be replenished by organic decomposition. If I have organic matter in my soil, that's going to replenish the availability of those nutrients. Okay? It's going to depend on the kind of organic matter. It's going to depend on the soil water content and temperature, climate again. Nutrients, some nutrients can be retained more tightly in the soil because of higher capacities of that soil to hold on to certain things. The classic example here is the phosphorus one. What happens when the pH of a system goes low? How does the phosphorus bind in those systems? Aluminum and iron. Okay? As the systems become at a higher pH, phosphoruses are bound to calcium. Maximum availability of phosphorus tends to be around 6.5 because you're minimizing calcium and aluminum and iron. Okay? So that's a property of retention. That's a property of the soil that has that's a retention property of the soil that holds on to that. In this case, phosphorus. Does that make sense to everybody? Yeah? Ish, kind of? So, how do we in fact figure out what availability is? And there's basically 2 methods. One is a soil test, okay, and there are 3 factors that control this availability, and then a plant response. So let's start with the soil tests. What are the factors that are going to control that soil test? Now, if I'm doing a soil test, and I take a soil and I measure the nutrient availability, and there's a lot of different types of tests that we use. Okay? Most of the tests that we use basically are mimicking the ability of a plant to extract nutrients. It's not a test, and we talked about this in lab. Okay? It's not a test that's going to take a soil and basically completely digest it and tell me exactly what everything is in the soil. What it's going to do is it's going to be a test that can extract or mimic the ability of a plant to extract. This is not a total disillusionment of the soil. Okay? This is basically a wash of the soil. It's washing at a concentration that basically the plants would be able to wash it off. Does that make sense? Okay, so we need to have an understanding of what the concentration of the nutrients are in the solution, not in the total volume. Okay? We need to have an understanding, if this test, if it's going to be effective, needs to have an understanding about the speed of the replacement of that concentration in the soil. Okay? Now, it may, in fact, not be the test, but it may be interpretations based on the test. Now, when we did the submissions for Dillman Hill, part of that submission test was we want to know what these nutrients are, and this is the type of plant that we have. But we also gave a lot of information about the soil type and its location. Well, why did we do that? A lot of that had to do with this. I wanted to know what type of soil it is so that I have some sort of idea about, so I have some sort of idea about CEC and the ability of that system to exchange these nutrients on exchange complexes, right? If I just give you a measurement of this, I'm not going to have any idea about the measurement of replacement. And then finally, depending upon what kind of nutrient it is and in what kind of soil it is, I have an understanding of the mobility of that nutrient in that particular soil. Does that make sense to everybody? Questions? Should I restate that? Yes? Okay. So let's think about this. If I need to have a test that tells me about nutrient availability for my plants, or for whatever, I need to have a test that will mimic the plant's ability to extract that material. Does that make sense? Okay, and that's why I want to have sort of the concentration of the nutrients that are in the soil solution, but I also want to have an understanding about the speed of replacement. Okay, because if the plants can only extract a little bit, there's not going to be a lot of replacement. On the other hand, if the plants can extract a lot, there's going to be a lot of replacement because of the cation exchange capacity. Does that make sense? Also, potentially weathering reactions. Does that make sense? Now, for me to be able to give you that type of information, I can't strictly depend upon a test alone. I need to have some sort of understanding about what kind of plants you're growing, because I need to know what their lifestyle is, but also their ability to extract stuff. But I also need to know stuff about the soil that they're growing in. Okay, if I just have the liquid of the soil that they're growing in, it doesn't tell me anything about the surface areas of the soil that they're growing in, the solid part, right? That's why we ask questions about the soil. Does that make sense? Cool? All right. The second way we can basically figure out what's going on, or a sort of an assessment of nutrient availability, is basically the plant response. Okay, I can put a plant in the soil, okay, I can give it a various amount of nutrients, and I can look at its response to that soil and the amount of nutrients that I add. These are field trials. We do this all the time. And this is basically the basis for many of our interpretations of these soil tests. What we do is we go out in the field, we plant these things, we give it a variety of different nutrients in a variety of different soils. We see what this response is, so that when we get your soils or for anybody's soils, we have a number of where that nutrient is in the system, i.e. how much has been added to the system, and we have a measurement of what kind of soil it is, and we know what kind of plant is growing. So we just basically correlate the soil test with the response curve, and we can give you an idea about how much you have to add to get to this point. Does that make sense? Yes? Is it? I'm definitely going to start a coffee business in here. All right. And again, it's based on these three characteristics of availability. Sure.
So number 2 is speed of nutrient replenishment. And I know you say it's vague, but I mean, understanding the context in which the soils are in, how would you express that in terms of, would that be reflective of quantity of nutrients, or?
All right, so the question, let me restate the question. The question is, in reference to, how would we communicate this issue of speed for replenishment? What does that actually mean? How fast something is? Is that in essence what you're asking? Yeah. So if the plant starts sucking nutrients out of the soil solution, how fast is that response? It's a good thing I can return the answer, because I feel like I'm a really good Cornell student. I can spit back the question for you, but I don't necessarily know the answer on that one, because it's going to be really dependent upon the different types of nutrients. Some of them are going to be very quick, potassium, bang, okay? But on the other hand, potassium can also bind fairly well with things. Calcium is going to be something that's really, not calcium, phosphorus is going to be something that's really slow. And I think the speed has less to do with the actual time and more to do with the plant. If a plant, so if you think about replenishment, certain plants, all plants, they have certain stages of their life that they need to have certain nutrients. Now, in general, we all have sort of base requirements, but when they go into fluorescence or they seed set or something like that, during those periods of time, they have some sort of like peak need for certain nutrients. And that's really more about what we're trying to, it's not like base numbers that we're trying to give them. We're trying to give them the amount of nutrients they need at their stage of life.
So it'd be a timing.
So it's more a timing thing. So, right, so classic inorganic fertilizer amendments, it's you don't put all the fertilizer at the beginning of the season. You go in there at certain times of the year and you side dress. Okay, and you give the fertilizer when the plant needs it. And this is a really good example of when you really want to do this is sort of middle of the road type of fertilizer, but you really want to have some base level fertilizer in the soil. And this is classic organic sources. You know, if I put organic material in there, because organic matter decomposes slowly, I'm going to always have this baseline of nutrient availability. And then I got a certain crop in there that goes, at one point, it really needs a high demand. Well, my organic fertilizer is not going to hack it because it's just not releasing the amount of nutrients that plant needs at that particular time. And the argument there is that, well, then we go in there with some sort of quick release fertilizer. Now, that's an argument that, that there's a lot of room to argue about whether that's the way or not. But the reality is plants need to have some sort of base level, but then they also have to have these peak points in their lifestyle, in their nutrient demands. Okay? And that's where you sort of think about what this is. There is not any hard and fast rule that says nitrogen is going to be off at this, potassium is going to be at this, calcium is going to be at this. It really has to do with the type of soils that you have in the system and what's actually in the concentration of the soil solution, because it's an exchange between these two. Okay? Does that make sense to everybody? All right. So let's take the next step. If we're thinking about this availability, we need to start thinking about how the nutrients are getting into the system and how they're being lost from the system, okay? And this is where we get back to nutrient cycles and their losses, okay? Nutrient cycles in soils tend to be leaky, especially in highly fertile soils. Now think about that term for a second. We'll come back to that. Why are they especially leaky in highly fertile soils? Okay? Nutrient losses basically depend on four things. One is soil erosion. If I'm taking the soil away, I'm going to be taking the nutrients away. Certainly, if I have crops that are taking nutrients up, when I harvest that crop and take it someplace else, the nutrients are going to be going with it. I also have volatilization of gases and leaching. These two you guys are fairly familiar with. Okay? Now, let's go back to this idea about highly fertile soils tend to be more leaky. Okay? It's not that they are inherently losing more stuff because they're highly fertile. It has to do with the fact that if you think about where the pools of nutrients are, okay, if the pool of nutrient is in the soil, and the soil has a lot of fertility in it versus a soil that doesn't have a lot of fertility, given the same amount of erosion from each one of those soils, which one's going to be losing more nutrients? The one that has more fertility, right? If I lose a pound of soil from an acre of a highly fertile soil versus a a low fertility soil, I take a pound of soil off of each one of them, there's going to be more nutrients in the high fertility soil than the low fertility soil. Therefore, it is more leaky. Okay? The percentage of the entire system may not be any different. I may be losing 10% in both cases, but the absolute amount is going to be different. Does that make sense? I'm not even seeing. Yes? Yeah? Okay. All right, so let's talk a little bit about each one of those. Erosion basically removes the phosphorus and the nitrogen in the solids materials, okay? But we're also going to see more erosion when we have enhanced. It's going to be enhanced when we have bare soil, okay? If I don't have something that's holding the soil in place, I'm going to have more erosion, okay? If I have more erosion, I'm going to have more nutrient loss, okay? Because of the nutrients, where this solid material is going, it's basically going down into streams and into lakes. This basically causes eutrophication downstream, so I see algal growth farther down in the system, or in lakes, in essence. Okay? Crop removal, this is necessary if we're cropping something. We're going to have to harvest. Okay? And if we harvest, we're basically taking it with us. The N and K are basically removed the most from this system, but we can minimize some of this loss by returning crop residues to the soil. If we're just taking the seed and leaving the rest behind, that rest, the biomass of the rest, is going to have NPK in it as well, and that's going to go back into the soil as that material decomposes. Okay? Gaseous loss, this has to do with the nitrogen cycle predominantly, okay, but it can also have to do with sulfur. Okay? Basically, when you're looking at the ammonification, once that amine group from the crop residue or the organic matter decomposes and is put into an ammonia, ammonia is a gas, so it can literally volatilize from the system. Okay? Certainly when you burn stuff, you're going to have volatilization as well. Okay? And also potentially when the system starts going anaerobic, okay, if I start having something like denitrification, okay, I'm taking it from nitrate and putting it into N2, the nitrate is an available form. Yes, it can be leached, but as it is converted into N2 gas, it becomes volatile, which means it can be lost from the system. Make sense? Okay, leaching is the next one. Basically, this is possible loss for all soluble nutrients. If it is soluble, it will move from the water. And if the water is moving out of the soil system where the crop is growing, it's going to be taking those nutrients with them. Make sense? Okay, it's most significant when we're talking about things like nitrogen, sulfate, and potassium. Phosphorus can also be lost, but again, you have to think about this phosphorus. It binds very tightly, depending upon the pH, with aluminum and iron at the low pHs, calcium at the high pHs, and actually organic matter in the mid-ranges. Okay? We can minimize this loss. Actually, we can minimize most of these losses by doing the right thing management-wise, but when we're talking about leaching, we can minimize this loss by fertilizing at the right time and in the right amount. Why am I going to put excess nitrogen on the system when most of that's going to be leached away? Now, the reason I'm putting nitrogen on the system is for my plants, so I want to put it in a form that the plants can optimize getting and minimize its loss. Okay? Classic example there would be that slide that I showed you about rice. Okay? Putting in the different types of nitrogen, ammonium versus nitrate, into different parts of the system to minimize its loss from the system. Right? Keeping the nitrate in the aerobic zone, keeping the ammonia in the anaerobic zone. Yes, they're going to migrate, but it gives it one more step of the nitrogen cycle and the ability of one more, or slows down its loss and maximizes the plant's ability to capture it before it's lost. Does that make sense? Does that, you guys remember that slide? Yeah? Okay. This is all premised on one hypothesis. Well, maybe not premise, but one hypothesis sort of holds a lot of these things together. And this hypothesis is that healthy crops use nutrients better. Now let's think about this. Why do we think, I mean, if we're trying to minimize loss from the system, we want to really maximize, one of the things that we want to do is maximize the nutrient uptake into the plants, right? Because the more the plants are taking up, the less is leaving the system. Now granted, we're going to harvest, but potentially we're putting the residue back. Okay? So why would healthy crops, healthy plants, use those nutrients better or prevent or reduce the amount of loss from the system, with the exception of the harvest? Think about it. Why would plants that are healthy versus unhealthy plants go?
Well, if they're healthy, they'll be producing more biomass, so they'll be using more nutrients and less will be available to be leached.
Exactly. So if they're healthy, they're going to be producing more biomass. And if they're producing more biomass, they're going to be harvesting more nutrients. They're harvesting more nutrients, there's less of it to be lost through volatilization and leaching. More biomass is also going to anchor more of the soil, so I'm going to lose less of my soil through erosion. I'm going to capture most of it in biomass, which means some of it's going to be lost through crop harvesting, but if I put that residue back in the system, I'm basically putting it into that organic matter pool that's going to go right back into the soil. Does that make sense? Question?
How does that work in a closed system as far as leaching goes? So if you have potted plants, what's the predominant loss?
Closed, well, I understand the question of the closed system. I wouldn't actually call potted plants a closed system, but yeah, I understand the question there. So if we're having a sort of a greenhouse operation or a potted plant system, how would we reduce some of this? Well, certainly a potted plant system, you'd have a lot less erosion because you have a capture field, okay? But potentially material can be going out the bottom of the hole. Now that bottom of the hole is where you're going to see a lot of the leaching. Now how would you minimize that? Well, you could have better control of how much water is going into the system, or and more importantly, the nutrients that are in that, okay? Now those nutrients can be coming in as soluble fertilizer, but those nutrients can also be being solubilized in that soil media and being lost from the system. So a lot of control is right there. How you're delivering that water and in what amount. I'm sure most of you guys, if we think about these from pots sense, house pots, people that are growing house plants and things like that versus greenhouse plants. In many greenhouse systems, we just sort of excess water and let them come out the bottom because we have sort of the saucer is really the base of the greenhouse, the pad, okay? But often you see with people that have house plants, we see the salts actually build up in those pots over time. That's when you start seeing the white coatings everywhere. That's the salts building up. The reason that we're not seeing the leaching in that system is we are actually seeing leaching. What's happening is the water is going down and sitting in the pot, and then as the plant respires, it's bringing that water back up into the system. So in essence, we're creating, I mean, it's not truly an arid system, but we're looking at evapotranspiration bringing that water back up. Now, the transpiration isn't going to get rid of the salts, and so in that scenario, we're actually seeing a buildup. Okay? It's not truly a closed system, but really a lot of that has to do with how we're adding the water and what's in that water and how that water is interacting with the soil media. Does that make sense? Okay. Any other questions about that? We feel good about this one? No? Yeah? Okay. So this actually sort of segues right back into the pot experiment, but managing these plant nutrients, you sort of have to think about, well, where are we getting these nutrients? Okay? Certainly we can get them from organic sources, but we also can get them from inorganic sources, and we can get them from manufacturer sources versus natural sources. Okay? Fertilizer manufacture and use does not create nutrients. Okay? They're basically harvesting and concentrating. Okay? Now, when you say manufacturing here, most of us think of sort of the petrochemical industry, but you can certainly imagine this from sort of an organic waste management system as well. Vermicompost is a really good example of this. Composting in general is a really good example of this. When we went out to the waste facility, the waste compost waste facility, you guys remember the size of those windrows when the stuff came in? They were rather large. You know, within 6 months, they were rather small. Well, what's going on? We're blowing off a lot of CO2 is being blown off. In essence, we're concentrating material. Now, granted, there's ammonia and there's other things that are being blown off as well. But in general, we're looking at a concentration. Okay? We then take that concentrated material and then put it back on our fields. The same thing is happening with the petrochemical system, you know, the industry. We're looking at basically taking some material and concentrating it and then transferring it and putting it on our fields. Okay? Animal manure, green manure, compost, they contain nutrients that are taken from the soil. Inorganic fertilizer are basically mining enriched deposits. It's all about concentration of those nutrients. Okay? Therefore, fertilizer, in essence, is a non-renewable resource, in a sense, because in all cases, we're basically mining. Now, there is one exception, but in a sense, this is also a non-renewable resource, and that has to do with nitrogen. With nitrogen fixation, basically, we're going to be taking nitrogen from gas, and using biological chemical fixation, we basically take that nitrogen and convert it into ammonium or ammonia or NH2, some variant of that. Okay? But in all of those cases, there is a large energy cost, whether it's heat or whether it's ATP. Question?
What exactly is green manure?
Green manure is basically a cover crop or something like that that I'm cutting and throwing into the system. It's not literally green manure. Yeah. Okay. Okay. Questions? No? Okay. So let's talk a little bit about where these nutrients come from. Okay. The first one is nitrogen, and we always start with, not always, generally we start with nitrogen because it's usually the one that's most extensively needed or used. It's usually the smallest slat in the barrel. Okay. It's also the one, besides carbon, hydrogen, and water, that is used in most abundance. Okay? Nitrogen deficiency is normal. Okay? It's common and it's normal. And plants are used to this. Okay? Few soils can sustain multiple harvesting without some sort of nitrogen amendment. The crops are just sucking it up too much and taking too much of it out. Okay? Phosphorus deficiencies are pretty common there as well. In fact, I'm not sure if this is correct at this point, but about 70% of agricultural soils, not in New York State, but about 70% of agricultural soils are deficit in phosphorus for plant uptake for some period of time. Okay, questions on that one? I thought I saw a hand. No? Potassium, sulfur, zinc deficiencies are common, but they're probably, even with these deficiencies, they're probably not the limiting nutrient. Usually it's usually nitrogen and in some cases phosphorus. Okay? Oh, I thought I had another slide here. Oh, I do. I skipped sorry. I put the slide in the wrong spot. Other nutrients, iron, boron, molybdenum, manganese, copper, manganese, magnesium and manganese. Deficiencies are less common, but they are out there. Okay? Chlorine, cobalt, and sodium, these are really rare deficiencies, but they do happen as well. And then calcium, its deficiency is rare, but calcium's kind of interesting. When calcium is in excess, calcium can actually suppress other problems. Obviously, acidity, I mean, if you start throwing calcium in the system, you're going to be raising the acidity. It's basically a liming reaction. But you also can potentially be dealing with reducing issues of sodicity and salinity. This is where my slide was supposed to be. We're going to skip over. The transition here was basically, okay, well, we know what these nutrients are. How do we, in fact, make decisions about fertility? Okay? Well, let's take a look at a number of different crops. Okay, and this is a wheat, an alfalfa, and a tomatoes, sort of a variety of different crops here. The take-home story, in a sense, is from a farmer's perspective, is how much yield are they going to be taking off that farm? Now, this is probably a low yield for grain at this time, but just imagine that these are the numbers. Okay? Now, from a fertility management sense, what do these numbers actually mean? Well, when I harvest this 6 tons or this 20 tons or this 20 tons, I'm going to be taking nutrients with me. Okay? Now, if I'm taking a wheat, I got 6 tons, I'm basically taking kilograms per hectare, 120 kilograms of nitrogen, 30 in phosphorus, 30 of potassium, 25 calcium, 15 magnesium, and 5 sulfur. So in essence, this is removal, this is loss of nutrients from my system. So you have to think about what you're growing to give you an idea about the yield, and that yield, what does that yield actually reflect? Okay, now from a farmer's perspective, think about the application rates. Now these are not the exact same, and they're sort of out of order here. Okay, so field crop we can think of as a wheat grain, vegetables we can think of as tomato, and alfalfa is alfalfa. Okay, this is the yield. This is what I'm losing. Now from a farmer's perspective, I need to make that up somehow, a grower's perspective. Well, depending upon my crops, this is what I'm going to apply. Again, kilograms per hectare. Field crops, about 100 kilograms of nitrogen, 7 phosphorus, 4 potassium. That seems to be less. Why?
Because some of the residue is staying.
Some of the residue is staying, but these numbers reflect what's leaving. It's not the entire, it's not the entire plant, it's just what's leaving the field.
Those nutrients are still in the soil, but they won't become available at the rate that you need to crop again, so you have to add some to supplement what's coming into the soil solution naturally.
In many cases, that's going to be the answer. So the answer was, those nutrients are still in the soil, but they're not necessarily at the rates that are available, or they're not necessarily at the rates that the plants need. Is that basically what you said? Okay. And part of that is correct. But part of that also has to do with what other type of organisms are in the system and how is this material getting into the system. Okay. If there's organic matter in the system, there's still stuff there. If there's minerals in the system, there's still nutrients there. There's still stuff there. Okay? But I need to be able to supply the nutrients based on the lifestyle of my plant. Okay? And a good example of that one is actually looking at alfalfa. If we look at alfalfa, nitrogen application rates, alfalfa leaving the system, look how much nitrogen is leaving the system. Yet, me as a farmer, am I applying any nitrogen to my alfalfa crop? Now that has to do with my crop. What is my crop? It's alfalfa. What has alfalfa the ability to do? Or not necessarily alfalfa, but something that's in a symbiotic relationship with alfalfa. Nitrogen fixation. Put alfalfa into the system. I don't have to add any more nitrogen. If there's no nitrogen there, it's not a big deal because they can do nitrogen fixation. Alfalfa will go into my plant, and I don't have to worry about it. Does that make sense? So thinking about what your crop is, what you're harvesting, and what's leaving the system basically is telling the farmers what I need to put in the system based upon what crop I'm using. Makes sense? Yeah? Okay. So let's go back to these fertilizers again. Okay, where do these fertilizers come from? Okay, there's natural sources as well as manufactured sources. Natural sources can be both organic as well as inorganic. Organic sources are often what we think of as biomass type of amendments, manures, mulches, composts, that type of thing. But we can also have inorganic sources that are also natural. Example here is rock phosphate. It's not like we're manufacturing rock phosphate. This is a geological material. Okay? Now we mine it and we can process it to make it more concentrated, as we talked about before, but it's still a natural source. It is inorganic though. Okay, we also have, hey, what's going on here? We also can have manufactured materials, and nitrogen is the classic one of this case. And in soils, plants, animals, ultimately comes from the atmosphere. So when we're manufacturing it, we're basically taking it from the atmosphere, and this is the Haber process. Okay, this Haber process, from a manufactured sense, we're basically taking N2 gas with hydrogen, we're putting it over a catalyst under heat, and we basically make ammonia. Okay, the heat and energy here basically comes from coal, petroleum, any kind of hydrocarbon type of thing, anything to get energy. Okay, we can then take this and under pressure with low temperature basically make ammonia. Okay, and we can take this, or say liquid anhydrous ammonia. So we basically take this ammonia and concentrate it. It's not we're making. This is making the ammonia. This is concentrating it. Okay. Worldwide, nitrogen fixation, about 50% of it's biological, about 50% of it is industrial. Other nitrogen fertilizers, so we take that ammonia, throw it under oxygen, we can basically make nitrate, turn it liquid, Okay, a lot easier to transport than a gas. Okay, if we wanted to concentrate it, we can put it with ammonia, we can put it with lime, we can put it with calcium, make a urea, we can actually put it with phosphate, make an ammonium phosphate, all different kinds of things. The truth is, all of these are basically easier to handle. All right, all of those are, I don't, that just happened? Did I totally miss something while you guys look at the screen and I was talking? All right, whatever. Luckily, we only have 5 minutes left. All of these basically can get concentrated, and they're all generally easier to handle than ammonium. Okay, ammonia's gas, i.e. that's the big issue there. All right, question.
So they actually make ammonia, or they actually make urea from ammonia. They just don't use.
They don't use the urine, yeah. Because it's easier I hesitate to say this. I can tell you the party line. It's easier to handle and easier to deal with if you manufacture it rather than harvesting it from all these cows across the world or wherever sources. I'm not necessarily sure I buy that argument, but I do buy the argument the way the system is set up right now, because of logistics and supply lines and things like that. I do understand that argument. But I mean, we can get urea from natural sources, but right now the system's set up. Now I can complain. In fact, I do. I think this is wrong, but that's the way the system's set up right now. Okay. All right. I'm not sure this is going to work right now. So what I'd like to do is, I was going to do this tomorrow or not, on Friday, but maybe we'll do it right now. We've got 5 minutes. I'm not sure if I'm going to be able to break these guys up into groups, but I want you guys, we've been talking about fertilizer. Nutrients fertilizer and, okay, this are the 3 terms that I want to connect. Now, there is no way we're going to be able to do this in 5 minutes. This is a lot of stuff. Okay, there are a lot of connections here. Okay, but what I'd like us to do for the next 5 minutes, and I want you guys each to do this by yourselves. We're not going to break you guys up into groups. It's going to waste too much time. Think about it by yourselves. Pick one of these connections. Nutrients to fertilizer, fertilizer to management, management to nutrients. Pick one of them. And I want you to come up with a list of the connections. A list, not an explanation, a list. Because what we're going to do on Friday, at the tail end of this lecture, that maybe we'll get or not, at the tail end of it, what we're going to do is we're going to throw these 3 terms up again, and we're going to put them on each one of these boards, and we're going to come up with the complete list, and then I'm going to break you guys up into groups. And rather than 5 minutes, I'm going to give you guys about 10, 15 minutes. You guys are going to be telling me a story. Okay, so let's start right now. Let's make this list. It will make Friday a lot easier.
So we're not turning this.
You're not turning this in, okay? You guys are not giving it to me. You are hanging on to it, and you're bringing it back on Friday. Okay? So pick one of these pairs. Just one. Don't do all of them. Just pick one of these pairs. Make the connections. Go. You got 3 minutes. Okay, we got a list. Okay, I want you guys to bring that in on Friday. Okay, I have 2 more slides that I want to hit before I let you guys go free. This was where we were. The last sentence I basically said was, because of this transformation from that ammonium, we're basically making these things much more easier to handle. A liquid is a lot easier to handle than a gas. Okay, and also it affects, in a sense, it's not just sort of transportation, but it also has a lot to do with how you actually apply it. You have to have specialized equipment, and we'll talk about this later, I think on Friday. You also have to have specialized equipment to get gas in your soil. It's not like you just spread gas. And when I talk about gas, I'm not talking like gasoline. I'm talking like gas. Okay? You can't just sort of spread gas on your soil because it will just volatilize off. Okay? So you actually have to have very specialized equipment to be able to use gaseous forms. Okay? Phosphorus fertilizer, the main industrial source is rock phosphate, which is basically apatite. It's mostly like Florida and things like that, where we're literally mining rock. Okay? They then process this to grinding, which is what we basically see as a rock phosphate. We don't get the rock itself. They grind it up. But we also can mix it with sulfuric acid, making superphosphate, or we can put phosphoric acid in and basically turn it into triple superphosphate. I can never barely say that. But basically what you're looking at here is sort of a 3% phosphorus content. This one says 9%, and this one you're looking at like 20% phosphorus content in that material. Okay, so that would be an NPK of potassium fertilizers. These are another primarily a mined material. We're looking at sedimentary deposits. Sulfur, we're basically looking at gypsum, which is a mined material. This is fairly abundant. This is also mined from sedimentary material. The other good thing about this is you're getting multiple nutrients on this one, and it's fairly soluble. Okay. Elemental sulfur is another source. This is fairly insoluble. It's an oxide, but in soil, when you put it into with oxygen, it basically turns into a sulfate. Now the problem with this is that you potentially will get the hydrogen with it. This is a lot of what you see with, not the elemental sulfur, but with the sulfides, this is a lot of what you see with acid mine spoils, as well as dredged material. The sulfur comes up as a reduced sulfur, gets in the presence of oxygen, it becomes oxidized, and it basically turns into sulfuric acid. Okay? That sulfuric acid will drop the pH of your soils pretty precipitously. Now, this can actually be a management goal, though. If you have a really high pH soil, you might in fact want to use this sulfur, not necessarily for the sulfur, but for the protons to drop the pH of your soils to a more ideal zone for nutrient availability. Does that make sense? Yes, kind of? All right. Then phosphorus. We also have organic fertilizers. This is the last slide before I'm going to set you guys free. Organic fertilizers basically are derived from plants. Ultimately, they're derived from plants, even if you're looking at a bone meal or a blood meal or something like that, because those animals are basically surviving off of plant life. Okay, so unprocessed, obviously, things like leaves, clippings, things like that. We also have animal processed stuff where they're taking the plant material and we're looking at things like manure, bone meal, seaweed sludge, all that kind of stuff. And then we also have microbial processed stuff where we're looking at composts. Okay, vermicompost is sort of a blend between the two. Okay, the vermicompost is an animal, but there's a microbial linkage there. Okay, does this make sense? Okay, the take-home message from this slide and some of the previous slides is fertilizer management and fertilizer manufacturing is really all about concentration and moving those different pools to where you want.